Okay, sir. We'll start at
We'll wait
2:40,
for 5
sir.
minutes. Yeah, we'll start by 45.
2:45, sir. Okay.
That 2:45, yeah.
Okay, sir.
Okay, ma'am. So around 40 members are there. 40, 41 are there. So
Shall we start, sir?
yeah.
Good afternoon, all. I once again welcome you all to this afternoon session of day 5 of our refresher course. It's my pleasure to welcome our respected principal, Dr. M. Ramanathan, sir, Professor and Head, Department of Pharmacology, PhD College of Pharmacy, to take over the session. Welcome, sir,
Okay,
and
ma'am.
over to you, sir.
Okay, thank you. So dear participants, so we have asked the delegates, anyone of them are interested to present their research proposal, what they have learned it or anything they have it, so that we thought we'll do an evaluation with our team and then give them the feedback. Two of the delegates given their shown their interest yesterday, and many students have submitted their paper presentations also. So we thought should be a research proposal and a new research thought. So we thought it's okay not to, because when we allow one student, then another 12, 13 are there. So there'll be some time constraints will be there. So because of that, we restrict to the two faculties, and they withdrawn it. I don't know the reason why they have withdrawn it, and might be the time constraints, or they are not very comfortable to present to the podium, or some, I don't know the reason. It's fine, no problem. But then what I thought is some of the points which you people may not be known, or which people may not be understood, which we learn it through experience. See, any learning has to come through experience. And this experience, whatever you learn it through the experience, may not be there in the textbooks. That's the reason why it is always, we say that any research activities to be associated with the lab which has been well established, because they have a trend and they know what is to be done and how it will be done and what is the methodology will be adapted, and they will be having optimized protocols, procedures, equipments, and they also working on some certain hypothesis which they know it will function well. And also you can take up some new a new hypothesis or new molecules or new pathways. There we can explore it, and you'll get a better results, and that can become a new topic for the next research. Because any research you do it, any research we do it, my feel is it has to, any research if you do it, it will give some open up some questions and give you some and put some notes. That means it will create some more questions. It should create some questions. That should be the research. And that point, that whatever may be your observations, which is not being answered, will become a hypothesis, and you will design an experiment again on those hypotheses, and you will proceed. And over 5 to 10 years of time or 15 years of time, you can see a lot of work your lab would have been done it on a particular area. Then you'll become an expert. Then you can call it that your lab is so-and-so lab, and you are working in so-and-so area, and you become an expertise in that line, and you can start reviewing the articles. Then you get a lot many funding projects and everything. So it's a long-term process. So my feel is, if anybody wants to come to research, you cannot expect any short-term goals. It is a long-term process. You need patience. Patiently you have to wait, and you have to put constant effort and learn it. And whenever there is a new technology comes, again you learn it and you unlearn it, which are you learned it earlier. That is the way how it has been processed. Okay. So with this, I will share some of the slides which you might fill it, which is not there in the text, but I have written it through my experience, which might benefit you. Okay. So is the slides are visible?
Yes, sir.
Okay. See, when you write a proposal, when somebody comes and meet me, I always ask them whether it is feasible within your 6 months time or 1 year time or 3 years time. If any given PhD work, see, initial 1.5 years, you do a lot of bench works, sorry, a lot of homeworks. You do this, you do literature review, you frame hypothesis, you design experiments, you take ethical committee clearance and everything. It takes a minimum 1 year. Then another 1 year, you will spend it on optimization of your work. Then once you have done your optimization, the last 1 year only your real productive year comes. And the 4th year, you'll be consolidating the data and start publishing it. And 5th, 4.5 years or 5 years, you may get your degree. So that is your regular pattern when somebody wants to do PhD. And that's the reason why when you submit a proposal, we aim it for 3 years time. So that timeline, whether it has been feasible, that you should always work it out. You can think many things, be more ambitious, but whether it has been able to complete within that period or not, that is very, very important. And second thing is, you have written so many objectives, 8 objectives, 10 objectives, 5 objectives, 3 objectives. Whether those objectives are able to complete it with your proposed methodology and that within the time frame, that is also very, very important. You might have some 10 points, 10 objectives, but some of the objectives may not reflect it in your methodology. And when you commit into an activity chart plan every 5 months or 6 months, what is your plan, that should reflect it. Then again, about the start and end date, when you are started, when you are going to end your work, that is very important. Generally, when we say a starting date is for the project is not the sanctioning date, the date when you received the finance, the DD or check or the direct beneficiary transfer, DBT, that will be the date of commencement of the project. That is very, very important, because based upon this only, you're going to submit your auditing statement. So many people will miss this date. So when you started your work, that's the reason I always insist all my students, you should have a diary. Even you're not done any work, you write not done any work. Suppose if it is a Sunday, just you write it a Sunday, no problem. So that you'll have some continuity. And the feasibility of the proposal judged. The feasibility means again, whether you are capable and whether your lab is capable and your expertise are okay. And again, the time frame, whether it is possible to complete all the work which you have proposed it. Suppose if somebody have started identification of plant, then extraction, then identification, isolation of the compounds, then bioactivity fractionations of the compounds, and followed by in vitro and in vivo assay, then formulation development, it is highly hypothetical. If I want to isolate a compound, I need minimum 3 years to 4 years of time. That too in microgram quantity. So definitely I will not fund those projects. It's waste of solvents and chemicals. Instead of that, I know most of the phytoconstants are known. I will straight away go to the phytochemical and then do the job straight away from the chemistry data bank or phytochemistry data bank. Why I should go for the molecules? So one of the parameters for evaluation is this time activity plan. That is, you should always remember. And timeline is not a fixed document. It differs from person to person, project to project. Because suppose if you are doing some clinical trials or clinical investigations, you are written some 100 patients will be recruited. So is it possible from that center what you propose to get 100 patients or not? That is again very, very important. Okay. So these are all some of the points you should always remember when you write a proposal, or even can be M.Pharm project or a PhD project or your postdoctoral research, anything, but you think it of how it should be. And if you're doing animal experimentation, and if you're a fresh graduates doing the job, and I know how much you can do it within the frame of 6 months. And because you should get the ethical clearance and you should know how to handle the animal, you optimize your methodology, then you start working. So maximum 2 to 3 parameters you will complete it. That's enough for your M.Pharm. Otherwise, you work for more than 1 year, no problem. So next is when you return a proposal, what could be your outcome? What extra added informations you are going to give to the society? Don't say I'm going to find a chemical molecule, I'm going to patent it. Nobody is going to fund you. We know what is the importance of patent and how you are going to capitalize that patent into a financial, convert into financial benefits. People will know that, or how you are going to transfer the technology. It's all a big question mark. But whether it is really a new one, are you going to find new molecule, a new pathway, new mechanism, new mutations, or new indications, or you're repurposing your drug already been existing? So these are all some of the questions will be your outcome, which is based upon, again, your hypothesis and your objective. And if it is not matching it out, then your study will not have any significance at all. So here, definitely the evaluator will assess the significance of your research outcome. Suppose if you say you are going to develop deliver a drug molecule to a brain, or gene delivery, or you are going to produce a stem cells which are going to replace it for the islets of Langerhans or in the liver or in the brain, it's fine. But how we are going to do it, what is its application part, what is the translational value, all these things you should remember it, which might help you to get to a better funding. Okay. So research outcome is to be stated with very clear way, and not to give in a big paragraph. It should be given in a very, very nutshell. And people should understand. Suppose if your outcome, if your research is not in path, what is an alternative mechanism do you have it to make it complete? Because always I say, when your route 1 is not answering well, you should know what is the alternative route 2. When the route 2 is not answering, you should know what is the alternative route number 3. So then only you will be a successful person. Otherwise, you'll be sitting on the data and sitting on those experiments, and you'll never produce any data, and you'll be going to depression. So research is a unique one, which is not, again, I always say it's not a cup of tea for everybody. It always differs from person to person. And for this, you need a clear, focused activity. That is most important. This is a cartoon just for your fun. So I just is written for a manuscript, but I take it for a proposal. So you are going to get your proposal accepted, but nobody is going to support you. Nobody is going to support your proposal or even your paper for publications. They always see how to reject it, including me. If any project comes to me, I always see how to reject that particular project. What could be the basic ground for that? So that is the mindset of all the evaluators. So don't think that everybody will accept your paper. See, for example, I reviewed one article. See, it looks good. See, that particular person wants to develop a aerosol substance containing coagulants. His objective is when somebody met with an accident, to stop the bleeding, he wants to spray that aerosol so that the bleeding will arrest immediately. Here the question is, see, when you take a coagulation, it is both external as well as internal process. And when you have a severe bleeding and hemorrhage, it is very tough to stop the bleeding because it's a big wound, big hit is there. Even a drug cannot stop the coagulation process because a big injury. So in that process, how we are going to help and coagulants, how to going to help it in the clotting process. So that fundamental itself is getting wrong. So there is no need to go further about the thesis, directly reject there itself and submit the report, finished. So just only an abstract and objective, just studied, finished. So every angle will think it of how to reject a project, how to not allow it, how not to allow the things. This slide is not changing. Hello?
Sir, it is not changing, sir. It's in timeline itself.
1 minute. Is it changing now?
No, sir.
Why it is not changing? Now?
It's not changing, sir. It's okay, sir.
Is it changing?
Yes, sir.
Okay. So this is the one which I am telling about the cartoon. Okay. The persons, and this is about the research outcome of the timeline, and this is about the paper acceptance. Okay. So apart from this, you have to you have to submit some appendices, which is essential, like certificates from the head of the institution. It's always better to have a proper channel, and ethical committee documents, if it is involves humans as well as animal, ethical committee document has to be submitted with valid dates, and biodata of the entire team, PI, co-PI, their team updated is required. And this previous project's completion details is very important if you have any ongoing project, because what happens when you feed your name in the data bank of the government bodies, they will know all your history, whether you submitted the proposal elsewhere or the same proposal, you change the title and submit to this process, or whether you have completed the project and again you're resubmitting with the title change. So everything will be known, because it's not possible for us to see, but for them, for the ministry, it is easy for them to see. So it is always better to state the previous project's completion details and their UC and SE of the previous project, because again now it is on the online mode, PFMS. And this, where you again scroll it as a PI of Ramanathan, if I put, I then come to know all my ongoing projects, whether it is been whether it has been approved, and all my UCSC have been approved or not, everything been seen. And suppose if you involve clinical trials, clinical subjects, you should have an informed consent and data collection sheets. And if you have an industry, then the industry should have to match it out their certification process. These are all basic mandatory that will be checked by the DST or DBT or ICMR, CSIR or any funding agencies. If any of these documents is not been improperly submitted, then they will not process your applications at all. So this is again very, very important. And make sure that these details have been submitted without any delay. And again, certain undertaking also to be submitted that I have not given it here, but it's a part of the application itself. So undertaking by the PI. Okay, so these are all very important material you should remember. Then if you want to apply to a funding bodies, so the skills what you require is your subject knowledge and your research skill. That's why I say don't jump. See, whether fortunately or unfortunately, right from my beginning, 91 onwards, I am working only in neuroscience area. Recently, I started because of the collaborator, I started working for past 6-7 years on the prostate cancer or the breast cancer, where we started working on 7-8 years. That too involving the similar pathways, because there also we are working on neurogenesis, cell proliferations, neuronal proliferations. Here also we are working on the apoptotic mechanism. So I thought it's fine. No problem. I can work out on it. So don't change oftenly your area of research. And what is your depth of knowledge in a particular area is very, very important. And if you have a critical thinking and analytical ability, that is very, very important. Why I'm getting that answer? Why I'm not getting those answers? If I'm getting the answers, what could be the reason? How can I elucidate those unknown mechanisms? Don't say that. That some mechanism which you'll be knowing it. You should know it. Try to read more and more. Think, and broad thinking is very much needed. If you don't think broadly and wider thoughts and lot of logical thinking, if you don't put it, then you cannot make out any data. If I give it to you, the excellent data, if I give it to you, that will not fetch you any meaningful results. Data are data, but your skill, thinking skill, or your analytical skill, logical thinking skill will differentiate from person to person. And if you have that ability, it will make you a good researcher. And if I want to have that ability, then I should know how to apply what I studied in my UG and PG levels, even in my journal. That's the reason I say journal clubs are very, very important for the PG students. And if the teachers are very good, they'll make you to analyze and make you to understand. They change your perception at all with a particular paper if you interact with them. Okay, so that skill is very much needed when you want to be a researcher. I'd say it's a good researcher if you want to be I'm not telling extraordinary, then you can become a Nobel Prize winner, but I'm telling only for a researcher. And of course, you should know to communicate your data in a proper way, in a writing skill or podium presentations or poster presentations in a good language. You should know how to present it in a neat way. Sometimes, even the project may be a little bit here and there, but the way which they have written it, if it is very impressive, so those projects also will get funded without any hesitation. Okay. So remember, if you want to be a researcher, as far as I am concerned, these things are very, very essential. You can think it of any of your good teachers or good professors whom you have learned it. Definitely, they'll be having some of these extraordinary skills which might help them, which might have supported them to be a good researcher. Next is common mistakes to be avoided. Submitting a lengthy proposal without any meaningful answers. People will get bored. Covering too much on research ground, like literature review only, or not citing major works in the literature review. Suppose if you are working in the area of neuroinflammation, I should know who are all the expertise working in that area in India, in abroad, I should not miss those papers. That means I am hiding the knowledge. Too much focus on minor issues, poor grammar and careless writing, no proofreading and revision. Some of the proposals what we receive is they simply cut and paste. They have written one object, but methodology will be something else because they clubbed 2-3 projects here and there and presented it in the, because most of the time what happens, the funding agencies will give you only 1 month time. By the time you see the notifications, it will be almost 2 weeks is left out. So last 10 days, you'll be rushing up. So you do many mistakes. So don't do that job. Be prepared and do it meticulously and you submit. And if you have not done it with your whole heart, better don't do it. It's waste of time. And you are wasting somebody's timing also, because it is going to submit to someone else. And I already told 4-stage review is going to happen, and everybody time you are killing it. So better don't do that job. So take care of these smaller mistakes, then we do it. Okay. So when it comes for the review, it's a time for review. So if somebody sends me a proposal for correction, so how I will do my review, how I do the review process. So these are the say 10 points or 8 points, 8 points, which is very crucial for me to analyze, for anybody to analysis. Essentiality or originality. Essentiality is the word which is used in the human ethics, and originality we use it in the research process. So whether your work is original. And many times it is tough to get a highly novel projects. But somewhere, something you try to answer it, that can become originality. So that small change, a small deviation. If, for example, we did 2 projects with ER receptors. A simple hypothesis is ER alpha and beta receptors. ER beta is pathologically expressed and having a less binding packets compared to the ER alpha receptors. And whether to develop a molecule in that or not, just we started. Then we thought ER antagonist should be a drug of choice. But later we concluded that forecast ER modulators. ER modulators will be the better drug of choice to counteract the ER receptor mediated prognosis of the cell growth. And that we did it the DST as a 2 terms project. It was very successful projects, and both of them are, one fellow is in industry, another fellow is in US. A simple point only, ER alpha and beta receptor, the line is ER beta is more expressed in the pathogenesis. So whether that can be a target, and if it is a target, how we can explore it better, and how we can make it into a molecule of success. So that is the way which we have done it. Similarly, if you take, we have done it with GSK also. That is again a big project. GSK, alpha, beta, both are there. Alpha is known for glucose, insulin, glucose, metabolism, and other things, and cellular growth. Beta is more of apoptosis and beta-catenin pathway mechanisms. And again, GSK3 beta mediated inflammatory markers disturbances, how we can control, whether we can have it in a, whether we can explore it in the inflammatory markers. Just only that small line. GSK3 beta is more of beta-catenin mediated responses and outputting responses. So what happens to the upstream, what happens to the downstream responses, how to modulate. We never gone for any new molecule with the GSK3 beta work. That got big funding. And another one is about the JNK3 receptors expression in the brain. And that again, that gentleman finished his PhD and now he's in working as a medical institute. Okay. So a simple one line, other on the story. So how that story you're going to write it, how you are going to elaborate it into a good research with your supporting methodology and experimentation and your literature review with your theory. So that is the level of these three sentences. So originality, how it is going to give an impact, and whether we have research questions like hypothesis, whether it has been framed or not. And when you have these three, whether it has delineate my methodology, and based upon this, the outcome has been correlated well or not. Then of course, the sample size, experimental like replicates are very important for the human data because the power calculations are very essential for human data. And again, the feasibility, you should remember when you use the clinical samples. And again, preliminary data is advantage. So Dr. Vadivaran also told about this preliminary data. So when you have a preliminary data and to support your hypothesis, if it is working well, then it is easily get funded. And of course, you should have a very clear analytical plan also. So this is the very, overall perception, how any individual is going to evaluate your work without any, complicated. These are the points they're going to work it out. But when we start reading your paper, research work, we can make it very clear whether you have experienced, you are written very logically, and whether you're able to do it or not. Okay. And research capacity, budget, timeline, these are all already we have talked more. And recommendation to the authors, if it is needed, then the score will be given. The research capacity, I told about your expertise, individual expertise. Then budget, if you ask more for money, for equipment and less for functional, nobody is going to give more budget for your non-recurring because it has become an asset. So don't ask for any lab establishment. Then strength and weakness of the proposal, your hypothesis framing and your methodology, your expertise, these are all your strength and weakness. And if you are not, but the project is okay, but not been up to the mark, but still it is fundable means sometimes I received sometime because I have not done any evaluation so far, but it's my perception I'm telling you. Because don't take it as I'm an evaluator. No, I'm not an evaluator. But sometimes I receive recommendations from the expertise that I have to change it. So every proposals I used to receive a recommendation. So once if I submit the recommendation, then they'll approve it and then they will give me the fundings. Okay. So remember, these are the points which you should always think it of before submission of your work. So in the conclusion of this 5 days event, what I want to stress upon is, but I don't know how many of you really listen to it because we are taking much of pain to call everybody and everybody spared their time, valuable time. All are good researchers in a different institute. And I hope at least out of 50, 10 to 15% or 20% got benefited. So to conclude it, research proposal writing is an art. It comes through practice. You have to write and write and write. And if you are not ready to write, you have to sit at least 2 to 3 hours per day for a month. Then only you'll get a proper proposal. And don't hesitate to present it to somebody. Don't think that they will steal your thought. No. The knowledge what you have gained is yours. Nobody can steal it. But you can give it. Even if you give it, how much they will take it, that is another question. So I have my own knowledge. I can give it to my students. Everything, whatever I know, I can tell. But how much they take it from me, that is a big question. So don't hesitate. Don't hesitate to share, to discuss, to present before submitting to a forum whether I have done it rightly or wrong. Then only you will learn. I always insist, try to accept criticism. If you don't accept the criticism, you will never learn in your life. Because my teachers are taught me like this only. Novelty is very important. Understand the thirst area. So many times when they announce the funding, they always give some thirst areas. So that thirst area, you should have an expertise. Many times I used to see that if I read it very quickly, and if I feel I can, I will. Otherwise, simply I'll ignore it. I'll never break my head there. Because once if you know it is not going to work out well, then why to break your head on that? Why to waste your energy? Because it is gone from your hand. It's not your area of research. Better you leave it. So working in depth in one core topic will be beneficial, that I have told many times. But dear delegates and students, nothing is impossible. Only your mindset should differ. Anything can achievable at any time, at anywhere. Only you need your mindset of doing a good research. So these are all some of the points which I thought to express it as a concluding remarks of this 5 days program. But I started with my failures. Now I say I can, okay, I am success. But still, best has to come. Thank you. Thank you for your patience hearing. Anything, if you want to ask me some questions, definitely I can answer you.
Participants, if you have any questions, kindly type and post it in chat box, or you can unmute yourself and call sir, ask directly to sir, which you can be clarified.
எல்லாரும் இருக்கிறாங்களா இல்ல ஆன் பண்ணி வச்சுட்டு போயிட்டாங்களா?
Sir, good afternoon, sir.
Yes.
Sir, for applying for this funding, we should have the publications on the area which we have proposed. Is it correct, sir?
You should have, yeah, definitely you need paper in the area of research.
If we are having a new idea, but we don't have the publication, so it won't be further processed, sir.
It is tough to get funding. Otherwise, you should have one co-PI.
Okay, sir.
Who can work on you, on your lab. See, how can you think, see, when you have a new thoughts and new idea, so you may not have that facilities in your lab, no?
Yes, sir.
So then how they will fund you? Otherwise, you should work on those line and for 1 or 2 years,
Okay.
see the service, have good papers, then you apply.
Okay.
Participants, any of the others need to be clarified with any questions?
Okay. So, Deepa Lakshmi, money to everyone, unable to unmute, sir. You can put it in the chat box, Deepa Lakshmi. So Deepa, you have some questions? Okay. Okay. So if there is no questions, who are sent to everyone. Sir, how to basic start research ideas, sir? See here, I'll give you one simple logic. Okay. I don't know how many of you will know that. This one I asked my students to do with my BPharm project. Take 1 gram of salt and 10 ml of water. Okay. You weigh 10 plus 1, 11 grams. Now you dissolve this 1 gram of salt into 10 ml of water. Theoretically, it should weigh, the salt solution should weigh how much? 11 grams. Is it weighing? Is it 11 grams or less? Any questions from anybody? சொன்னது புரிஞ்சுச்சா? பூவரசன்
புரியுது சார்.
புரியுதா? சரி, இப்ப அந்த உப்பு தண்ணி 11 கிராம் கம்மியா இருக்குமா கூட இருக்குமா? So it will be less. So now comes why. Other research.
Okay, sir.
புரிஞ்சுச்சா?
புரியுது சார்.
Right. In the presentation, do you have any answer for that? Why? I don't know which batch, your batch or your junior batch I did. Senior batch I did. I don't know this. Learn through simple experiments.
Yes, it is because like water is of less weight than the salt, and
No.
when we dissolve in that,
See,
sir.
the salt will become ionized
Okay, sir. Okay, sir.
and it's gone into a solution.
Okay, sir.
Specific gravity will increase and not the weight.
Okay, sir. Okay, sir. Okay.
Understood that?
Yes, sir. Yes, sir. Okay.
That's the simple logic why people will not sink in the Dead Sea or Red Sea somewhere. Maybe
Okay.
salt. Okay. Just for fun, I'm telling. These are the way how you learn science. It's not a big deal. Huh? Learn through fun. Cherry. Any other question? Okay. If no other questions, I thank all the participants and other members also for their wonderful 5 days program conducted. And 3:30, Narayan sir will be joining. Once that has been over, then we'll be submitting the question. We'll be submitting the questions and also the feedback forms. Kindly submit and kindly fill and submit it. Thank you. Over to Kritika.
As the learning is a never-ending process, we are learning from you every day, sir. We have learned from you, learning now, and will learn forever. Thank you, sir, for sharing your fruitful experience as how a researcher should be, like patient, updated, and highly committed. It's really an informative session for all the upcoming young researchers, either MPharm or PhD or any other scientist. So thank you so much, sir, for spending your valuable time for us. Thank you so much, sir.
Thank you, ma. Narayan sir vandangla.
Yes, sir. Yes, join now, sir. So participants, now we'll have a session from Nagarajan, sir. Good afternoon, sir.
Good afternoon, madam.
Welcome, sir.
Sir.
And over to you, sir.
Thank you.
Good afternoon, sir. Good afternoon, sir. How are you, sir?
Good afternoon. Fine, sir. Thank you, sir.
Okay.
Thank you. Sorry for
Yes,
the
sir. Please,
trouble in
yes,
the
sir.
morning.
Just now I completed mine. First you start, sir. Yeah, please.
Yeah, I was listening to your presentation, sir.
Okay,
One
okay.
more refreshing my my knowledge also. Thank you, sir.
No, sir. You are great.
Okay, sir.
Thank you, sir.
Let's start the session, sir. Okay, sir. Thank you. Okay, good afternoon, everyone. We are starting the session once more. So the title of the presentation is Protocol Preparation in Life Sciences Project Proposals. So I am very happy and honored to be part of this event because I enjoyed my stay at PSG for more than almost 8 years under the guidance of Principal Ramanathan, sir. Thank you so much, sir, for the opportunity once more. So the presentation will cover a brief explanation about what research is and defining research protocol and explaining the purpose of writing the protocols and describing the main elements of research protocol, what are the components, and I will also list the criteria for research protocol review. Once the protocol is written, either yourself or your peers or the superiors or the review committee members, they can review that. So what are the criteria on which it is going to be reviewed? And I have given also some examples about titles. The title plays a major role. Some good titles and objectives from the public domain, and a few methodologies and protocols, and some outcome also from the public domain I have collected for information. And lastly, some important points to consider before preparing for your writing a project proposal. I think Sir has already given you the tips, so I don't know whether this will add value. Let's see how it is going to be. So introduction, it is about what is research? It is a planned investigation that uses scientific methodology. So scientific methodology is going to be utilized, and it is an organized or planned activity. So we start by making synthesizing a small compound and then purifying and going for its characterization, and then evaluating its other properties. Ram Praveen.
Say M minus, anything, any problem?
Yes, sir.
Yes, sir, you are.
Yes, sir. I think I would like to share the screen from that side, sir, your side, sir. I can use the mic. So please share the screen from your side. The PDF file I sent in the morning, no, Ram Praveen, please.
I'll
Okay, sir.
Kritika.
Okay, sir.
Ma Kritika, open பண்ணி வச்சிங்களா?
Okay, sir. Okay, sir. Just a second, sir.
Okay, ma'am. Please do that. Huh? Okay,
Okay, sir.
yeah, it's fine. I am able to see the screen. So I was talking about the introduction, which is a planned investigation that uses scientific methodology. It is systematic investigation, we told. And then, yeah, that is the introduction. Go to the next slide, please. Yeah. So the research protocol should answer study questions. It should achieve the study objectives. Whatever objectives we have, it should be able to answer the objectives. And it should be a doable experiment. Whatever protocols or the experiments we are planning that are shown in the proposal, we should be able to execute it with the available facilities. And also it should be reproducible, so that anybody across the globe, whoever wants to do that, they will be able to repeat that experiment. And it should be also convincingly show the importance of the research. That is the, for example, it is the showcasing the capabilities of the research investigators. Some new techniques may be used for synthesis, like a microwave synthesis or a combinatorial synthesis, or maybe the use of advanced animal models for investigating breast cancer or other cancer types. So that will be done by our investigators' results. Whatever results they are obtaining, will be able to do that. Next, please. So the elements of the research protocol should contain these parameters, like a project title. It is very important. Project summary. Project summary is usually given at the end of the investigation. So at the beginning of submission, we will be preparing an abstract and project description. This is actually the body of the project work. That also contains several events, that is protocol. And we should also capture the national status. It is very, very critical to collect national status, because whoever is involved in this particular area of research in India, we should know. It may be used maybe for networking, or maybe to clarifying our queries, or maybe they may become potential reviewers for your proposal. So that should be actually captured. International status, this gives an idea about who are the prominent researchers across the globe. And rationale, very important. We may be attempting to develop a new method of synthesis and evaluate for maybe Alzheimer's disease. That rationale should be highly important. It has to be very clearly mentioned. And novelty. Novelty is also very important. Then dissemination and results and publication. That is at the end of the results or during the process of the investigation. Ethical considerations. I think Sir has already given you the information about how to get and how to properly document the ethical clearance. Timelines and milestones. It is another factor that has to be shown, because the project is normally funded for 3 years. So right from procuring the raw material or collecting the plant sample, or maybe from plant from a wild species, the timeline for collection, processing, and creating animal models, all those factors have to be clearly mentioned under the timeline. And milestones, at what period of time you will get a critical results, that has to be highlighted. And problems anticipated. Sometimes we are aiming at making something new. During the process, we may encounter problems, or we may actually predict this may work or may not work. So that problematic areas we can highlight at the beginning itself. Budget
budget is another critical factor because they normally fund it based on the experience of the researcher. For example, if a new researcher is applying, you should aim to apply for a project for maybe around 30 lakhs or less. If you apply for a 75 lakh grant at the beginning itself, it gets a chance of rejection because unless or otherwise it is a highly important area of research, but still it will have to cross 2 reviews, especially the financial committee. There will be 2 levels of scrutiny. So better apply for a grant at a lower level, 30 lakhs or 25 lakhs, get that grant first, and then apply for next grant, maybe slightly higher value. So that's why budget is important. So during the budget preparation, manpower cost it has to be collected, and equipment cost. Whatever equipments are already available in the facility, in the institution, we should not ask for the same instrument. That also has to be very critically analyzed. Whatever is not available and very frequently used equipments you can procure. Very rarely you use it for like an NMR. If you want to use buy it may not be given because the NMR you take only for selected compounds, synthesized compounds. So you can outsource the facility. Whereas creating an animal facility like a microscope or something which is very specific, you can buy. References, clear references have to be mentioned, and the research team, composition of the team, based on the expertise of the investigators, you have to choose. Like chemistry, you have to take person with a chemistry background, pharmacology, who is having experience in pharmacology, animal handling, cell-based analysis. And if it is a formulation related work, maybe person with pharmaceutics experience can be taken. Annexes, that is the other part which will actually document the biodata of investigators, key contributions of the investigators in that area, or if any mentors you are trying to mention their biodata, all those things you can actually incorporate. So this is the composition of the research proposal and protocols. So can you please move on to the next? Yeah. The project title, as I mentioned, it is very important. So that gives you preview of the project. It is like looking at the person who is very smiling and you encounter that person, you feel very happy, right? So like that, the title itself will give an impression. If you write a good title, that reviewers will feel, okay, this title is impressive, like that. So we have to choose right words, and it has to be concise. But sometimes it can be detailed also. Depending on what you want to investigate, if you cannot put it in a very concise manner, you can include additional details like a detailed title. And it should accurately express and to reflect the study. Whatever study we are undertaking, it should be actually captured in the title itself. And it should describe what has to be done. And in case of clinical trials, for example, the study population, maybe like diabetic people, or you are investigating memory enhancement, you have to choose the right population for that. Some inclusion exclusion criteria can be put under the variables of interest. And some important or interesting titles I have shown there: isolation, molecular characterization, and biological evaluation of anti-diabetic principles from a few Indian medicinal plants. This is one title which captures choice of an Indian medicinal plant, active compound, anti-diabetic activity, isolation, characterization, and biological activity evaluation. So this biological evaluation, actually this title doesn't capture what kind of anti-diabetic activity they are going to mention. Either it may be alpha glucosidase activity or maybe other activities that is not shown there, but still it covers most of the important parameters for that. Next title, new generation low molecular weight heparin analogues as anti-thrombotic agents and their preclinical evaluation. This is almost like a complete project description coming under the title itself. It is a low molecular weight heparin new analogues, new generation. So that means they are trying to address the existing heparin analogues, their problems that will be addressed with this new generation compound. And anti-thrombotic agents, that actually says that it is going to be evaluated for anti-thrombotic activity, and it is under the preclinical level itself. So they mention their preclinical evaluation. So it is like more or less complete information is provided in the title itself. Next one, nano drug polymeric nanostructures for treatment of microbial infection. It is for antimicrobial activity. It is nanostructure having a polymer plus a nano drug. So that is also covered there. Next one, novel nanostructured enabled drug delivery system for the treatment of tauopathy in animal models. So this is a very interesting title because it talks about Alzheimer's, not directly, but it says the problem in Alzheimer's like a tauopathy, and they are going to evaluate using animal models. And it is a nano-structured drug delivery system. So that means they are going to develop a nano-structured drug delivery system, NDDS, and for Alzheimer's using animal models. So that means these are some of the interesting titles. Apart from this, you can also look at certain titles like targeted delivery. They will say, for example, transferrin targeted liposome decorated doxorubicin, something like that they will put. That means they are trying to address the problem from the title itself. They will get an idea. So like that, there are certain interesting titles you have to coin using keywords. So this is an example. Next is the project summary or abstract. It gives an outline of the entire project. So you have to write about around 300 words. It should stand alone. That means it should contain almost all the information in a very brief manner. It should clearly state the research problem. Whatever title we have given, that title can be expanded to give these details like a research problem, main research question, or the hypothesis. What is the hypothesis you are going to give? Like the previous titles, nanostructured drug delivery system for treating tauopathy. So that is the hypothesis. So that means already existing drugs, whatever acetylcholinesterase compounds or other compounds, they do not have the capacity to penetrate into the blood-brain barrier or give a beneficial effect. So they want to address that. So that is the hypothesis they are making it. So that hypothesis should be mentioned here. And the justification for research. So how it will differ from existing studies. So that has to be highlighted here. And the research objectives. What is your objective? By doing a novel drug delivery system, you want to investigate for tauopathy using animal models. That is what is your objective. And the study design, what will be done? By whom? To who? When? How? And for how long? So this is actually study design usually is related to clinical trial activities. So when it is an in vitro or in vivo studies, you can accordingly modify the content. So it is about what kind of animal models you will use, what is the measurement you are going to make, like your animals response after a stimuli, all those things can be captured. And you have to also mention what is the expected outcome. Compared to the existing drugs, this new novel drug delivery system for tauopathy will be superior. That is what you are expecting as an outcome. That has to be mentioned there. And there should be some rationale that we will come later. And anticipated problems. Suppose you are trying to do certain studies with animals, which cannot, these properties cannot be studied in using in vitro or cell-based assays, then there is a difficulty creating that particular animal model, creating that particular disease. This is very, very critical. So that is to be created. And the description of the project, it should actually identify the research problem. What is the research problem? Same thing, what was mentioned in the title, you can use it, like a novel drug delivery system for tauopathy. Research problem, justification, rationale. That rationale is very important because they will always look at what is rationale. Why you want to do this? That has to be clearly mentioned. Then coming to the literature search, the literature search should be very thorough. I will come to that also later, how to do that. And study goals and objectives. Research question or hypothesis and the study design. And research methodology. Research methodology is actually the workflow or work plan and the expected outcomes. So this is actually going to describe the project. Next, please. So research problem identification. how to identify the research problem. What is the problem? Recently, we all know that there is a COVID-19 spread. So that can be a potential problem for addressing that issue. So that title can be taken. And you have to see that whether it has a significance, especially because it is affecting so much people throughout the world, and frequency of occurrence or public health interest. This is actually a public health interest, and also like a cholera or other kind of diseases, viral spread. Are the causes of the problem known? If you know the reason for the spread, you can address that. You can explain, this is the reason I have taken this as my research topic. And solvability of the problem. Can we solve this problem? That has to be assessed so that you can give a solution for that. And a need for research to address the problem. Any knowledge gap. There may be some information already available, but it is not complete. So by doing some research work, you will be able to fill that gap. That is what has to be identified as a research problem. Next. So justification or rationale. You have to argue why the research should be done. That is what is the rationale. Somebody will ask you, I want to go to Canada. You have to justify why you want to go to Canada. You have to say that lifestyle is good, very safe country. Why you don't want to go to countries like Australia or other places. You have to say that I want to go to Canada because of this problem and this advantage and that advantage. That is what you have to argue to say why you have taken this as a research problem. And you have to also answer these following questions. What knowledge information will it provide? Whether you will get a new knowledge, whether you will get a new drug so that the population can be cured from this deadly disease. And what are the plans to disseminate results? How you are going to publish the result, or you want to take it further, maybe to take it for clinical trial, or if it is a very important discovery you have made, you want to patent it. So that also can be mentioned. I want to patent this product so that the country will benefit from this. And how will the results be applied and who will benefit? The entire society will benefit. The country will also get benefited. Like if you're generating a new vaccine for COVID, if it is chosen as a problem, it will actually benefit the entire country. So that is how you have to justify. So you have to make sure that the research topic you have chosen is unique. You have reasons to justify that it has to be conducted. Next. So this is an example of targeting BRK positive breast cancer, small molecule kinase inhibitors. So what these people have introduced to the topic is 80% of the breast cancer overexpresses kinase, breast tumor kinase, BRK protein, which is having various oncogenic roles in breast cancer cell proliferation, survival, and migration. So that means it is actually a cause of spreading the disease very fast. But it has not been explored. That is what they say. BRK inhibition have yet to be explored as possible therapeutic tools. It has been evaluated under biochemical investigation. Several biochemists have investigated the role of this particular kinase, but no effort has been done to develop a drug for this. So these people want to develop a drug through this process. So that is what they highlight. And a small molecule inhibitor, they want to synthesize a small molecular inhibitor so that it can selectively inhibit. This is how you have to highlight, selectively inhibit the kinase activity of BRK, and its downstream signaling results in significant anti-proliferation potency. So that means their entire outcome is captured here. So that problem, when they started the problem, they just mentioned that there is an unmet need. That means there is no cure for this particular situation through this process. So they want to develop a new molecule. So that they have done. They have developed this compound and evaluated, and they got some interesting results. That is what they have done. Next, please. Similarly, development in PARP inhibitors in triple negative breast cancer. So what are the triple negative breast cancer features and what are the role of PARP inhibitors? They have given this as their rationale for choosing this as a project title. So they have given the challenges like triple negative breast cancer. It is actually identifying a subset with the homologous recombination deficiency, all those problems are there. And it has to be identified. And PARP inhibitors, using this, maybe they will be able to develop an intravenous or a PO dose, or schedule treatment versus continuous. So instead of continuous treatment, you can give an incremental treatment. That is what they wanted to evaluate PARP inhibitors as compounds for treating triple negative breast cancer. Next, please. Literature review. As I mentioned, literature review is very, very critical. You have to give, if you look at the bottom of the slide, systematic review using databases like a PubMed, Science Direct, a Google Scholar. You have to use standard databases, not like a simple Google search. It will give you millions of hits which are not at all related to your topic of interest. So you have to do a thorough literature search using these tools. You have to also use Boolean operators like and, or, with, not. These are the Boolean operators. By using these Boolean operators, you will be able to filter the search results so that it will be narrowed down, so that you get a clear picture about your hypothesis or your topic of interest. You can also use PICO, that is a population intervention or context and outcome. PICO. PICO is a methodology usually used by experts for searching literature. So using these tools, you can do a literature search, which will give you a summary of the critical review of literatures on the research problem. Unless you do this systematic search, whatever search you do may not be sufficient. You may end up with too much of information, which is not at all going to be useful for writing your proposal. And it actually gives you the research problem into perspective, because it will narrow down the search results. You will get a clear picture about what is known, what is not done, what can be done. And it justifies the new research by identifying gaps. That is what I told you, identifying the gaps and weaknesses in existing literature. And using these tools, you can do a logical and well-organized, thorough literature search. Next, please. Study goals and objectives. You have to fix the study goals like aim, general objective, which actually informs broadly on what the research proposal will do. Study goal or objective. So it will give you a tentative idea about the project. An ideal study goal should be clear. It should state the purpose of the study, about what you want to really do that. And you should define what will be described and determined. What kind of activities you will measure, what kind of information you will derive from that results you have to mention. And to define, identify, compare, and when there are hypotheses, it should indicate what will be confirmed. The hypothesis, if you have written, that hypothesis will be confirmed is going to be your objective. Next. So continuing on that, the statements of the research questions should be captured there. You should formulate, before starting the research itself, you have to formulate the objective. After starting the work, you cannot formulate a hypothesis or objective, and indicate how the study will accomplish its goals. And you have to introduce the study design. The parameters that you want to evaluate have to be clearly mentioned during the preparation of the proposal under objectives, and should be simple, concise, and very specific. You cannot do every activity that is known for a particular compound. Depending on your activity that is going to be measured, you have to specify what are the measurements you will make. Next, please. So, for example, I have captured recent topics from the study goal and objectives. A general objective may be preclinical evaluation of small molecule LOX-L2 inhibitors in breast cancer. So, this is a new topic. LOX-L2 is a lysyl oxidase like 2. It is a member of the lysyl oxidase family of amine oxidases. It is actually involved in normal tissue development and homeostasis, and also onset and progression of solid tumors. So that means this is an important new avenue for breast cancer activity. So people started working at this particular target. So the objective, they have given preclinical evaluation of small molecule LOX-L2 inhibitors in breast cancer. So specific objective is to test anti-tumor properties of 2 generations of novel small molecule inhibitors in MDA-MB231 human model of breast cancer. So that is their specific objective. Overall objective, preclinical evaluation, small molecular inhibitors, specific 2 generation of LOXL2 inhibitors. They will also investigate, the objective is to investigate inhibition of LOXL2 activity and further growth of primary tumor, reduction in primary tumor angiogenesis. So they want to measure how this angiogenesis will be affected by these compounds. Through this investigation, it will provide first evidence of support the role of LOXL2 specific small molecule inhibitors as a potential therapy in breast cancer. So that is how the objective has to be framed. So look at this. It is a very, very interesting new topic. So much work has not been done on this. Someone is interested, you can look into this particular topic and see whether any new product, natural product can be used, or new synthetic compounds can be evaluated, or maybe repurposing of old compounds can be considered as inhibitors of this, provided you have access to the cell-based assays for this particular target. So this is an example, interesting example of a study goal and specific objectives. Next. So research question or hypothesis, how you frame it? A good research question should have a simple, clear and unambiguous hypothesis. Like in the previous slide, they have indicated LOXL2 inhibitors to treat breast cancer, right? It should be clear and unambiguous. They want to synthesize some compounds, evaluate further. It should be focused. Their interest is inhibition, realistic. They want to synthesize 2 generations of compounds, not 10 generations, right? 2 generations, realistic. Answerable, that means it clearly indicate what data will be needed to answer the question, how it will be collected. So this collection of data, that is actually the protocols or the experimental part which will be covered later. It is very interesting and logical if there are more than one question. And express as a question. So the question is whether these LOXL2 inhibitors will provide treatment option for breast cancer. That is their logical question. Okay. That is how the hypothesis is framed. Then when you want to frame the question or a hypothesis, like in the literature search, you can use PICO or spider terminologies, which will actually guide you to develop a hypothesis. PICO is a population. What is the population or the disease? What is intervention or issue means? What is the drug that you are going to give? And comparison, how it is comparable to existing drugs or in the context, and what is the outcome? That is PICO. So using these 4 keywords, you can frame your question and do a search in PubMed or Google Scholar or Science Direct using Boolean operators. So it is a 2-way approach where you use these terminologies, combine the keywords under this terminology and use Boolean operators, then get a very narrowed down results so that you will be able to get a good hypothesis for your research problem. Spider, again, the same kind of approach, same sample, phenomena of interest, design, evaluation and research, specific to qualitative. So like this, you have to frame questions, do a thorough literature search, a systematic search using Boolean operators, using these terminologies. It will actually help you to generate a question which can be framed into a hypothesis. So please remember, this point is very useful. Next. Again, coming to research question or hypothesis. So a declaration of the relationship between 2 or more variables, one being dependent. So if it is a compound and a disease, you will be able to compare the dependency between these 2 variables and tentatively predicts the research outcome. What is the outcome that has already been known? See, for example, curcumin and breast cancer, if you do a search, you will get n number of results. So if you use certain terminologies according to the PICO method, you will be able to narrow down, maybe like reducing some toxicities or improving bioavailability. Like that, you will be able to narrow down the result. Tentatively predicts the research outcome. What results are already available, you will be able to get, and should clearly state the variables that may be likely associated and the population of interest. So that is actually interlinked between the PICO terminology and the Boolean operators. Okay. So based on this, you may say it is a null hypothesis. You already know it, there is no association between the variables. It is a null hypothesis. Alternative hypothesis, there is an association between the two variables. That means whatever you do, you have to also always relate these two factors. That is how it is an alternative hypothesis. Next, please. There are some examples which are not exactly related to pharmaceutical research. It is actually in the case of clinical applications. For example, the study purpose they have given. Purpose of the study is to determine if there are differences in pain control with nurse versus patient administered analgesia following surgery. So the patient himself is taking the analgesia, whereas in the other case, the nurse is administering it. What is the difference? Is there a difference in pain control? The question, you can frame it, does the administration of analgesia by nurses versus by patients themselves affect pain intensity during post-operative recovery? It is actually reframed, right? Finally, you refine that into a hypothesis. Patients who self-administered narcotics will be more satisfied than patients who receive narcotics administered by nurses. So this is the hypothesis. So you have to do a research and find out whether this hypothesis is working, or from the population study, you can collect the data and prove whether this hypothesis is right or wrong, right? So you have a study purpose, you refine it into a question, that question can be refined further into a hypothesis. So how this is actually done? It is actually done through a systematic search using the terminologies like PICO and SPIDER, and also the Boolean operators, using valid databases like Science Direct or PubMed. You have to do that. Okay, go to the next page. So this particular thing, need of nanomedicine for breast cancer therapy, shift from conventional to nanomedicine. That is their focus. So what they have done is, conventional chemotherapeutic agent, it is associated with many limitations. There are n number of limitations written one by one. It is having a rapid drug clearance. It is having poor selectivity and poor target-related delivery. All those problems are listed. So finally, what they want to do is, in order to overcome all these problems, we read this. Therefore, multifunctional smart nanoparticle conjugated with the targeting therapeutic fluorophore can cross different biological barriers, target and penetrate cancer cells by passive method known as EPR, enhanced permeability retention effect, and finally release drug in the cancer cells in a controlled manner. So finally, what they want to do is, because of all these problems, they want to develop a nano formulation. That's all, using a carrier. So that carrier will help them to deliver the drug to the cancer cell. It will overcome all the problems listed there. So this is their objective. How they came to this objective? Because they studied all these problems associated with the existing drugs. Based on those problems, they wanted to provide a single solution using multifunctional smart nanoparticle conjugated with targeting fluorophore compounds. That is what they are aiming at, and then they will study all these properties. Okay, so this is a kind of knowing the existing problems, you will be able to provide a solution for that. So this blue colored information is actually their hypothesis. Okay, next. Now if we look at this, it is actually giving the nanotherapeutic targets for triple negative breast cancer. They have listed down what are all the problems associated with that and how it can be addressed using different type of approaches. Targeted delivery, using different type of formulations, all those things. Can you go to the next slide, please? Okay. So using these things, you can actually design your study. The research question or hypothesis is actually important. Based on that, you compare with alternative study designs. You propose something and also propose some alternative study designs. You evaluate the feasibility, resource availability, timeline, ethical consideration, because suddenly you cannot administer a drug to a human being or an animal. So ethical clearance. So like this, these are all the factors you have to consider. So which will determine the credibility of the study. You also have to state and justify the study design. Using these factors, considering these factors, you have to propose a study design for your proposed work. Next, please. Next slide. Okay, research methodology. The methodology is actually the plan of work. What kind of activities, the sequence of events you do, all those things are captured. So it is most important aspect of a protocol and should be written in detail. That means it will cover the maximum pages in your proposal. So it provides details and justification of the techniques, procedures, which will be used to achieve your proposed objective. So that means it is related to your objective only. Whatever objective you have given, you have to provide information about the kind of activities you want to measure, and based on that, you have to plan your research methodology. Next, please. So you have to provide the information like variables, what kind of variables you want to use, like different animal models, study population or research settings, sampling. It is for the clinical trial, proposed intervention. It is also related to clinical aspects. I think we'll go to the next slide. Next slide, please. Yeah. So all procedures for data collection. You have to provide details about which instrument you are going to use and for what purpose you are going to use, pilot testing, recruitment of study participants for clinical trial, data quality control, and data management and analysis. What kind of statistical tool you are going to use, what kind of controls you are going to use, and clinical data, you have to code the data, monitoring and verification, computer software, statistical methods, sample size, and study power, significance, all those things like p-values, all those things have to be captured under the research methodology. Yeah. So this is an example of a research methodology given for a particular work. What they wanted to do is use a Transcriptional peptide. It is a pentapeptide. It is they call it as a double-edged sword in Alzheimer's disease. They want to evaluate this peptide, pentapeptide, to treat Alzheimer's disease. So the code is KLVFF. It is called Transcriptional peptide, which has been used as a beta sheet breaker to disrupt amyloid protein aggregation. That means it is used for Alzheimer's disease treatment. Some indications are there that they have got some data like it is able to break beta sheets. That's all. But what they want to say, emphasize here as their objective is mechanism underlying the transition behavior of peptides along with the molecular targets involved remains unexplored. That means there is no relationship between how this compound or the peptide is able to break the beta sheets and what is the molecular mechanism and the targets involved is not clearly mentioned. That is not available. So we want to evaluate this. So that is why they want to say that this forms the major focus of the present study. That is their objective, right? Then for that, they have given all the protocols, what kind of activities they will give. So they will cover the details under the materials, what kind of materials like the KLV peptide, other compounds for in vitro screening, and methods, different methods like cell culture and seeding. These are all standard procedures you can collect from literature and use it. And lactate dehydrogenase assay, there is a standard procedure for that, but they want to use this study, they mentioned it. But that means the study they have mentioned, all these are related to their objective. The measurement procedure may be already known, but they want to use this measurement to support their objective. So they want to use lactate dehydrogenase LDH assay, reactive or oxygen species determination ROS, intracellular glutathione level, thiabarbiturate acid reactive substance T-bars assay, imaging of cells using microscopes, gene expression studies in order to evaluate the changes, and superoxide dismutase activity SOD, caspase activity assay, tracker red staining, JC1 mitochondrial membrane potential, and finally statistical analysis. So that means these studies are very critical for meeting their objective, right? Okay, next. So you have got an objective. You propose several protocols to meet the objective through experiments. And what is the expected outcome you have to predict? What new evidence will be obtained from the study? Is there any new evidence? Because they have indicated that there is no molecular mechanism related to that particular pentapeptide in treating Alzheimer's disease. Similarly, they have to identify what kind of molecular mechanism is involved. So that will be captured in their protocol. Then outcome will be coming from their experimental data. And how will the evidence contribute to improvement in knowledge? So if they are able to prove that this is able to act through this particular molecular mechanism and this target, that is a new knowledge. That is how they will get an outcome. Who will benefit or use the findings? So this finding will be used by pharmaceutical companies because they will know that there are these pentapeptides are available. They can actually evaluate in animal models and then find out whether it is actually curing this Alzheimer's disease and any effect on healthcare, health system or policies. So that is at the extreme end of the investigation. Final benefit that is coming from as an outcome from the study. Okay, next please. So from the previous study, the pentapeptide they have used, what they have done, concentration dependent dual free radical property was observed. That is their outcome. One. Then activates cell proliferation scavenging gene AKT and mTOR. That is actually helping in the cell survival. So these are two major outcomes from their protocols which are related to their objectives. So the objective is giving them some options for protocol or experimental work. That experimental work gives some outcome which is actually related back to the objective. So they have also found out a higher concentration is toxic. That means they are giving some information that the dose has to be adjusted. Maybe you're using some animal models and other studies they want to evaluate. And then the other outcome they have got is it is actually transcriptional factors NRF2 SOD levels are regulated through factors like AP1 and NF-kappa B. So through inflammatory pathways, these factors, transcriptional factors are also affected. So that is by the use of this pentapeptide, they were able to show there is a new mechanism that is involved, molecular mechanism, and the targets also that has been identified for their activity. So that is the outcome from their experimental work. And they also say that it holds immense potential in designing these peptide-based therapeutic strategies in future. That means they have observed some toxicity with a higher dose. They propose that there is a possibility because this cannot be directly used as such. They want to optimize it. So they say that there is a immense potential for designing optimum compounds for treatment of Alzheimer's disease in future. That is the outcome and their suggestions for future work. Next, please. So then, as I mentioned, the protocols or the experimental procedures or the work plan differs or defined based on the objective of the work. For example, this particular title, a new approach to inhibiting triple negative breast cancer in vitro, ex vivo and in vivo angiogenesis effect of this particular compound from this venom. They have taken this particular venom. They wanted to evaluate this property to treat triple negative breast cancer activity. So their protocol, apart from all the simple materials, methods, and general instrumental requirements, they have specified very important key experimental parameters, the protocols. They will do cell viability by the MTT assay, proliferation assay, cell adhesion assay, cell cycle analysis, cell migration assay, and dosing of VEGF in the supernatant of this ex vivo angiogenesis assay, mouse aortic ring assay, characterization of co-culture in MD-AMB231 cells and angiogenic sprouting, co-culture invasion assays, chick fluoroelectronic membrane angiogenesis assay. So these assays or the protocols are very specific for their objective. So depending on your objective, you have to design or you have to propose, collect information about the experimental procedures and list it down under the methodology. The procedures are already available in the literature. Only thing, you are applying it to your objective. So that entire procedure has to be copied. And when you copy the procedures, you have to only write in present sentence. It is not like it was conducted, it was estimated. No, it will be done, it will be conducted, like that. You have to reframe it. So this is a reference from which this specific assays for this particular objective was taken. Similarly, we will go to the next slide. You also want to evaluate sustaining proliferative signaling and evading growth suppression, because they want to see the triple negative breast cancer, right? So they want to evaluate evading growth suppressions, metabolic activity like ATP production, all those things they want to evaluate. Protein content, membrane integrity, like I mentioned, mitochondrial membrane potential they will measure using dye exclusion assays. DNA synthesis, how it is affecting it. Impedance of a living cell, using a live cell you can measure this activity and see whether the spread is there or not. Live cell lapse imaging, colony formation assay. So these are all critical protocols or procedures you have to use pertaining to your objective. Unless or otherwise all these protocols are listed, it doesn't make it a complete study, right? Then resisting cell death, morphological changes, genome fragmentation, plasma membrane alterations, and detection of apoptosis regulators, detection of caspases, senescence, inducing angiogenesis, all these parameters they want to ask. So this is an exhaustive study. They will be having only one particular venom they have isolated from a particular organism, but they want to do a complete study. So this makes a complete package. So by looking at the experimental protocol and the link of these experiments towards their aim or objective, the reviewers will appreciate, okay, there is some logic. These people have given an exhaustive and a comprehensive list of experiments to support their objective or claim. Once they are able to get the result through these experiments, they will be able to categorically say that this is really effective for treating that particular disease. So that is how it has to be a convincing list of experiments. Whatever is not required, you don't have to do it. But what is essential has to be captured. It is a very, very robust and comprehensive list of experiments they have taken. In some cases, it may not be so much comprehensive also. Next, please. So there are some other experiments like activating invasion metastasis and the transfer migration, Boyden chamber, scratch assay, cell exclusion zone assays, capillary chambers. These are all new type of experiments which normally in a very basic pharmacology lab we may not be doing it. And these facilities may not be available also. We have to take help from somebody who is having that facility. That is why it is required to collect the national and international status of that particular research area. If somebody in Naipur, Mohali or Chandigarh is having that facility, you can send your sample and ask them to request them to conduct this experiment, provide you the data. So that is why the research providing national international status also is very vital in order to thoroughly study the parameters or complete the protocols. Genome instability and mutation, all these things have to be captured depending on the objective. It is not necessary for every pharmacology work we have to do all these experiments. Depending on the objective and how these protocols are related, you have to choose those experiments. Next, please. You also know about flow cytometry, karyotyping, array of comparative genomic hybridization. You can also do some in silico methods like computational genome-wide association studies, GWAS. Like I mentioned, glucose uptake can be used to measure the ATP, activity of rate-limiting glycolytic enzymes like ALT or other enzymes, fructokinase or other enzymes, oxygen consumption, glutathione, right? These are related to specific objectives. Next, please. And next comes the publication and study findings. I think Principal Sir has already given you about the publication ethics. You have to follow all those things. So these results can be published in an internal seminar, regular reporting to stakeholders. Like when you are funded by a funding agency like a DST or DBT, periodically you have to send the reports. Like every year you have to send the annual report. Publications, journal articles, reviews or book chapters, conference presentations, whether national or international, some exhibitions like the IPC or other conferences, outreach and public engagement. If it is an awareness kind of activity, like use of a vitamin for treating or improving the mental health or something, you can create public engagement events. Based on the objective and the outcome, you can go for any of these publication related activities. One thing to be cautioned is, if you have found out something very, very interesting and which is very unique, you are not supposed to reveal to any of the colleagues or publish in a conference, publish in a journal, so that it can be patented. If you want to patent your invention, it should never been published. It should never have been presented in a conference. So that has to be kept in mind. But these are all general activities so that you can publish your results, so that you get popularity among the peers, also reveal your interest to the society. Next, please. Ethical consideration. I think it is more related to your animal handling, human handling, like clinical trial activities. So that is not so much relevant for our in vitro studies or other activities. But if you want to take some patient-derived samples, like a TB or scabies kind of activities, you want to collect skin tissues, you have to go for ethical clearance. So that is also very important. Without that clearance and you get to collect sample, it is actually unlawful. Keep in mind. Next, please. So timeline. As I told you, timeline is important. It specifies the duration of the project. 3-year project. You have to use judiciously to complete the studies. It can be classified under preparatory stage or training of research, equipment procurement, pilot studies, data collection, analysis, report writing, milestones for long-term projects. If you want to continue the project, you can ask for an extension. So that can be based on the milestone level results. Next, please. So whatever problems you are anticipating, you can highlight it, how the obstacles should be overcome. Like in one of the slides, I have mentioned about the toxicity at a higher dose with the pentapeptide. So that has to be highlighted, so that whoever wants to continue on the work, they can reduce the dose and see how it can be optimized. Next, please. Budget. Budget is also very critical. Realistic budget should be mentioned. Too much or too little may lead to unsuccessful funding. As I mentioned, more than 30 lakhs for an initial application. First grant should be avoided. Try to apply below 30 lakhs. Don't ask for only 5 lakhs from a funding agency like DBT or DST. Whereas UGC gives minor projects like 2 lakhs. Even Tamil Nadu State Council for Science and Technology gives 2 to 3 lakhs for student as well as teacher projects that can be applied. Whereas if it is a funding agency like DBT, DST or Nano Mission, you can apply for 25 lakhs or so. And types of expenses, it is already classified under their funding agency norms itself. What is going to be supported, what is not going to be supported. You can look at that and accordingly use that. Next. References. All references information should be cited. Plagiarism has to be checked. You cannot copy paste. You have to rewrite it, understand, grab that information, try to put it in your own words. Formatting, whatever format they are asking, you have to use, especially DBT. they have a boxes. Inside the box, you have to put only the limited number of words. They have word counts. So please see that it is considered before preparing that proposal, and then use it. Cite relevant and current literatures. So use those important relevant references there, and all cited authors and text should be in the reference list. That should be there. Not cite the author, first author, and then write the journal article name. List the entire author name, and references should be cited and listed sequentially. Whatever format they are asking, it may be from the sequence means it is like 1, 2, 3, or normally in the proposal, they are not asking you to write 1, 2, 3, author and year. You have to write that in that alphabetical order. That has to be followed. Next, research team. Who are all the participants of the research team? Like chemistry, zootics, pharmacology, or maybe process development chemist or industrial partner. Those people, according to their contribution and their expertise, you have to use that. And their curriculum vitae, CV also has to be incorporated. And one more thing which has to be kept in mind, when you apply for DBT, they ask for the publication list of investigators with impact factor 1 and above. Whatever impact factor is less than 1, they are not asking you to cite that reference. So that means when you publish an article, before going for a publication, proposal submission, you should concentrate on publishing in a good impact journal. Try to publish in at least 1.5 to 2, so that after a few years, even if it comes down also, it will be standing around 1 or 1.2. So don't aim at pay and publish journals. Wait for some time, collect some more information, publish in a good impact journal, so that it will be useful when you apply for a funded project. Okay, next please. Annexes, it is related to clinical trials, instrumental thing, quotations. Like quotations, if you collect for that instrument which you want to procure, you have to keep that quotation in hand. You have to also collect what are all the already existing infrastructure available, and financial and insurance linked to other projects. If you already have some other projects which are running or completed, that list and their status, whether UC and reports have been submitted, the status, the project closure report, you have to keep all those things ready. Cover letters, signature from the head of the institution for authorizing you to undertake the study, providing you the facility to conduct the study. All those things also have to be kept ready. So don't go to the head of the institution in the last minute with your full proposal and ask for his approval. Give them sufficient time so that they know what you are preparing and when you are going to submit, what is the due date. If tomorrow is the last date and if we go to the head of the institution with the full proposal asking for his signature, it is very, very difficult to get that. Okay, so please see that that is not done. Keep everything in place before you go there. Keep on discussing with your peers and the head of the departments and the head of the institutions, so that they will guide you in a proper way, so that you don't lose that opportunity. Because many times they call for new proposals and give only 1 month time. By the time we start preparing, it will be 25 days over. So beforehand, you talk to the head of the departments and other authorities that you want to work on this, you want to submit it. Who are all the investigators you want to propose? Whether you want to take the principal investigator, co-investigator as your head of the department or head of the institution, you have to get their concurrence in advance, well in advance, so that there is no problem. So you can submit the proposals in proper time, so that you get a chance of getting evaluated with the funding agents. Okay, next. Next, general considerations, table of contents. Wherever possible, try to prepare graphical abstract. It may not be available through the portal to submit graphical abstract, but it is always good to have a graphical abstract to cover what are all the key factors involved, how you are going to address the problem, all those things, do that. And be consistent in formatting, font size, font style, margins, numbers, all things you have to be very, very careful. Grammar, make sure your proposals stand out. That means you need to make sure that it doesn't lose out based on these silly factors. Even though the concept is good, the proposal protocols are very good, and the methodology is very nice, but the formatting and other factors are not very good. The referees will not feel very happy, right? So they may make a comment, and accordingly you may lose the chance of getting the fund, right? So please see that these are minor things, but you should give more importance while submitting the proposals. Okay, next. So whenever there is a call for funding, look at the objective of the funding agency. What is that it is related to? It may be related to COVID. It may be related to anti-malarial activity. So depending on that interest of the funding agency, you have to prepare your proposals and follow the guidelines and instructions. And you have to show enthusiasm and commitment so that you will be successful in that. Okay, next. So source of research funding, it may be public, it may be government agencies, or some private agencies may also fund you for a specific activity. Depending on that, you can do that. International agencies also sometimes they offer funding. Maybe at a later stage of your career, you will be trying to apply for those fundings. But like okay. Next, please. So the proposal review criteria is actually, you can evaluate, your peers or the head of the institution can evaluate. They will look at, is the research question important? Is the study relevant to the funder's interest? What is the quality of the research design? Are the investigators competent enough to conduct the research? Is the research facility capable of conducting the research? So these are the things they will look at it. Based on that, they will give you ratings. Accordingly, the funding agency will give you fund for your proposed research. Next, please. These are other factors they will look at it, the budget and the facilities available, the reputation of the institute, and the writing of the proposal, whether it is clear and well written, all those things they will check. Next. So as I mentioned, they will ask you to submit for a specific call. For that, you can apply for that specific call. Otherwise, throughout the year, there are funding agencies accepting proposals like a DBT, even DST Nano Mission, it is accepting throughout the year. DRDO Life Sciences Board accepts proposals. Department of Atomic Energy is accepting proposals. So like that, depending on the funding agency's timeline and the requirement, you can apply. ICMR, sometimes they call for funding agent call. Sometimes the Ayush, very specifically they call for funding. Only in newspapers or other advertisements you get it. Whereas under NMPB, National Medicinal Plant Board, you can apply throughout the project. Through the year, you can apply for a project. Like that, you can check the timelines from the funding agency websites, and accordingly you can submit. Next, please. So I think if time is not sufficient to cover this, this is related to how you will have to understand the biology of the disease, and accordingly you have to plan your hypothesis, how you have to plan your protocols, how you have to justify the concept. All those things are captured in the subsequent slides. So I think we will maybe continue on another occasion.
Yes, sir, it will be better, sir. 4:45 already. Yeah, 4:45 already.
Yes, sir. We will close it.
Yeah, we can, anything is there important, you just share it, sir. Or we can go to the conclusion.
Okay, sir.
Yeah, we have to close it by
Yes, sir.
It will take, sir, for
Yes,
you.
yes, sir. I think that is all. Yeah, I think there is all what I wanted to convey, sir. This
Okay,
is
sir.
actually kind of a tips for them to start looking at. This can be shared also through email or whatever way you prefer, sir. No problem. Okay, so I will make a conclusion. So when you want to make your proposal, you have to plan your concept, right? Creating a concept starts from literature search. If you do a good literature search, you will be able to come up with good questions. From the questions, you will be able to generate a hypothesis. From the hypothesis, you can devise protocols or methods, research methodology. Then you can plan for budget, manpower requirements, instruments, and then fit into the funding agencies' formats, and then submit it, and then you will be able to succeed. To make good collaboration with people who are experts in certain areas. Only one thing I wanted to emphasize is most of the B-Form projects or M-Form projects we undertake. M-Form, I think it is little advanced. B-Form projects or other projects we take one particular compound or an extract and go for anti-diabetic activity using alpha amylase activity. We stop with that. But we should look at all the other parameters like ROS and other factors and bioavailability measurements. If possible, do a docking study, compare with a known drug. Like that, if you do a complete study, you will be able to publish in a good journal so that you get a good publication and a good impact factor, which will be helpful for you to apply for a project at a later stage. So I think if you follow these methodologies, you will be able to succeed. And I wish all the participants success in their efforts to create new proposals, and wish you all get good funding also. And thank you for the opportunity. Thank you so much, sir. Any questions? I'll be happy to answer. Kritika ma'am.
Participants, if you have any questions, please do post in chat box or unmute yourself and you can talk to sir directly.
In the meantime, you can scroll to the next pages, madam. I will see whether any particular page is of interest. I will just show it. Just go to the next pages one by one. Next. I think it is maybe 10, 15 slides later, you will find something. Go forward. Yeah, you can go to 2, 3 slides. You can skip. Next. Okay, stop there. Yeah, either the previous slide or this slide, you can look at it. Previous slide with the image, madam.
Is this okay, sir?
Previous one. Yeah, this will be fine. Yeah. So this particular slide actually contains, yeah, this is also fine. Next, where there are 2 boxes with the hexagons. Next. Okay, then that's fine. So if you look at it, there are several targets listed on the left-hand side with the blue boxes. On the right-hand side, their functional role is indicated. For example, ROS, it is related to NRF2 and GSH. It is a drug efflux. You can look at it like ABC transporters. Cell cycle, You can look at the cyclin kinases. Like this, there are related. So you may have to relate the functional activity to a particular target activity. That is how you plan your protocols or the experimental activities. So there are some other examples I have given in the subsequent slides, which can be used later. So I think with this, we will close the presentation. If any questions are there, I will answer it.
Any questions you want to ask with sir?
Participants, any questions? Thank you so much, sir. Your informations were very much informative and innovative to newcomers actually, who are going to start their research. And the protocol which has to be prepared was very, very briefly explained, which was very much informative. Thank you so much, sir. We are here to more.
Thank you, ma'am. Thank you, sir.
Thank you, sir. Thanks for your timing, sir. So we'll have a brief discussion.
Oh, welcome.
We'll have it, yeah.
Yes, sir. Yes, sir. Thank you, sir. Thank you so much.
Thank you, sir. Thank you.
Thank you. Thank you, sir. Thank you.
Ma'am,
Sir,
conclude
shall we
பண்ணிடுங்க,
close
ma'am.
this? Okay, sir. So now we have come to the end of our virtual refresher course on our scientific program on research project writing, bioethics, and scientific communication. First of all, we would like to thank our beloved principal, Dr. M. Ramanathan sir, for being the backbone of this program and for his immense support behind us for organizing the refresher program. We especially thank all the speakers for this valuable time and informative sessions provided to us. We also thank all the faculty from PhD College of Pharmacy and other institutions, research scholars and students for your kind cooperation and patience for the successful completion of this program. And the participants will be sending you the feedback and evaluation form. On the basis of evaluation, certificates will be sent to you. Thank you. Thank you, everyone.
So, dear participants, don't forget, submit your feedback form as well as the questionnaire. Being it's the refresher course, we are asked some 15 MCQs. You have to answer those MCQs and you have to submit back to us as a evaluation pattern. And those who are submitting the evaluation pattern questionnaires only will be given you the certificates. So, dear participants, don't forget, do that. Okay. Being that today, tomorrow's sessions have been covered today itself. So tomorrow we don't have any, because there is no presentations of today afternoon. So we, whatever the Saturday's event, which we have done it today itself. So hence tomorrow there will not be any sessions, but only we need is your feedback and the questionnaires form. So I thank all my colleagues as well as Tamil Nadu Engineering University, Chennai, for accrediting this program with this event with the credit hours of 30, credit points of 30. And also the other teachers from my sister institutions and from government as well as from the industry for giving their allotting their time and giving the lectures. So see you once again in such other forums. Thank you one and all. Thank you once again. We can close it. Any feedback, anybody wants to talk, please, you're welcome. Any participants? If no feedback, you can, then it's fine. We can close it down. Yeah.
Okay, sir. Thank you, sir.
யாராவது வருவாங்களாமா? வெயிட் பண்றதா? 5 minutes எல்லாம் க்ளோஸ் பண்ணிடலாமா?